So welcome everybody to the weekly seminar of the Working Group on Multiscale Modeling and Viral Pandemics. This group is, and the seminar is organized by James Glazier from Indiana University and myself, and then James Sluka is taking care of our website, and also Lorenzo and Joel have been kind enough to help with other organizational activities, including inviting speakers and setting up, maintaining sort of our schedule of this seminar. As usual, you can find us on multiple social venues, including our Slack channel, Twitter. We have a wiki page, also LinkedIn group, and on YouTube, where you can also find previous recordings of these seminar series. So please follow us on Twitter. Help us spread the word about the working group, as well as about the seminar series that go on every Thursday. Do we have any announcements from the group? Okay. The other announcement is this new paper that's been published very recently by Gary Ann and Chase Cockrell about the development of digital twins for drug discovery, published just last month, so feel free to check it out. Also, if anybody has any suggestions on potential speakers, please suggest them using this URL and Google form. That'll help us, continue the series and engage the community as much as possible. So for the upcoming schedule, we have Nancy Baker from the EPA, who will be presenting on August 11th. August 25th, we have Carolyn Cho from Merck. We are still short on a speaker on August 18th. So again, if you have any suggestions for your colleagues and friends whose presentation might be relevant to this group, and again, we're very inclusive, so it doesn't matter if they're purely computational, computer science, or on the wet lab side, or anywhere in between. Please help us fill that spot. So with that, it's my great pleasure to introduce and have Patrick Kinnunen from University of Michigan and Jennifer Linderman's lab to present his research on the analysis of heterogeneous single-cell kinase signaling dynamics. Thank you so much, Patrick, for joining us here. Really appreciate it, and looking forward to your presentation.
As you already heard, my name is Patrick Kinnunen. I'm in chemical engineering at the University of Michigan, and I'm officially in Jennifer Linderman's lab, where we study tuberculosis and also sort of cell signaling dynamics and receptor-ligand interactions. I also work really closely with Gary and Kathy Luker in radiology. They are experts in microscopy and live cell imaging, and they helped me take the picture that you see in the background of the slide. So their input in this talk has also been really invaluable. So first, I want to talk a little bit about cell signaling and just give an introduction to make sure everybody's on the same page with some of the terminology. So cells live in complex environments, and they have to constantly sense their environment. So there are these ligands floating outside of the cell, and typically how cells sense their environment is a ligand will bind to a receptor. Chemical messengers inside the cell will then relay that receptor's activation to somewhere else in the cell, for instance, to regulate a gene or to regulate metabolism. And in this way, cells can maintain homeostasis in changing environments, right? So crucial for cell survival. The chemical messengers we're going to focus on here are kinase proteins. Kinases pass phosphorylations around the cell, basically. So a kinase will get phosphorylated, and then it can phosphorylate other kinases downstream. So two kinases that are particularly important are ERK and AKT. They control proliferation, survival, and migration, and they're common cancer targets. So I study them, but a bunch of other people study them too, and you'll hear a lot about them in this talk. And I also just want to sort of mention the standard way to measure kinase activity, and that's using a Western blot. So Western blots are great, but unfortunately, they are bulk endpoint measurements. So they require measuring a population of cells instead of single cells, and they also measure endpoints. So you can't gather data in real time using them. So this is kind of a picture of a Western blot over here on the right, showing that when you give EGF, a growth factor, phospho-AKT turns on, you start to see a band. But again, we don't know anything about individual cells, and we don't know anything about the dynamics of this process. So next, I want to talk about why we might care about single cells and dynamics. So on this slide, I'm presenting some data from an older paper. It's from 1994. And in this paper, they were studying how the progenitor cells to neurons were regulated. And so what this research group did was they gave those progenitor cells either EGF or NGF. And so those are two different growth factors. What you see in this plot on the left is that those two different growth factors provoke different kinetics of this kinase ERK. So the blue, the NGF is a sustained response, whereas the red EGF is a transient response. And interestingly, when you give NGF and have a sustained response, you see that the cells differentiate into neurons. Whereas when you just give EGF and you have a transient response, they proliferate. And further research found that actually the difference in the dynamics of ERK activity was enough to change whether cells differentiate or proliferate. So dynamics are important. The other thing that's important is about having dynamic data, sort of real-time data, is that it can allow us to better untangle regulatory mechanisms. So you can imagine you might gather some single cell data for something like protein expression. And if you're just looking at endpoint data, let's say you run an experiment where you give some stimulus, stimulus A, and then you have another condition where you give stimulus A and then stimulus B. And you can see stimulus A gives you this distribution on top, and stimulus B gives you a distribution shifted up or more active. Unfortunately, what you don't know is how stimulus A and B interact. And so there's kind of two different ways. There's a million different ways they could be interacting, but at least two. So you could have this situation where cells that respond strongly to stimulus A also then respond strongly to stimulus B. And so in this case, the outliers would be at the top of both distributions. But another case could be that stimulus A sort of inhibits or prevents cells from responding to stimulus B. And so in that case, you would see that the top of the distribution after stimulus A would actually be at the bottom of stimulus B. And these are, obviously diametrically opposed interpretations of data that's really can only be revealed if you're looking at cells in real time as they respond to both stimuli. So on that note, I want to talk just a little bit about how we acquire dynamic single cell data. And there's kind of a general flow to this. So the first thing that we do is we stably express fluorescent reporters in cells. So here I'm showing a schematic of something called a kinase translocation reporter. When this kinase ERK activates, you'll see that ERK activation will cause the fluorescent signal to migrate from the nucleus into the cytoplasm. And this happens very quickly, and it's also reversible, which is great. So once we have these cells expressing this fluorescent protein, we can do live cell fluorescence microscopy. And basically, in this stage, we're collecting videos of the cells as they respond to some stimulus. We do automated image processing on those cells to extract time tracks, and I'll talk about the form the data takes in a little bit. But on the right here, I'm just showing 3 pictures of the same cells as they, before they're stimulated in this top picture, 5 minutes after EGF is given, and then 2 hours after EGF is given. So we can see in these 6 or so cells in the middle, they're all off. The ERK pathway is off in these cells at the start because all the signal is in the nucleus. Then we can see that when we give EGF, cells turn on strongly. So everything turns into a donut. And then what we can see is that 2 hours after we give EGF, we've got a cell like this one out here that is still active. It's still a donut. We've got a bunch of cells here in the middle that are kind of in between. And then there's this cell where the signal has all or almost all migrated back into the nucleus. So we can kind of start to see that heterogeneity. I want to just, again, to make sure everybody's on the same page, I want to talk about what our final data output is. And so again, we have these videos that we're taking. So it's not just one frame, but we're seeing this sort of throughout time. And basically what we're getting is just a matrix of data. So the way that we organize the data, we have a matrix where each row represents a cell. And each column represents a different time point. So if I'm looking at a single row across time, I'm seeing how one cell is behaving at different times. And if I look down, I'm seeing the distribution of cells that at a single time point. So I might have 3 cells that I'm sort of analyzing, and those might have 3 different sets of AKT and ERK data. And obviously, if I have 3 cells, I can just look at some line plots. But usually we have hundreds, if not thousands, of cells, and so we like to organize the data in what's called a kymograph or a color map. And so this is just that matrix that I talked about presented as a color map. So each row is a cell, each column is a time, and then the color represents the intensity of signal at that time point. In this experiment, we have paired AKT and ERK traces, and about 180 minutes into the experiment, we stimulated the cells with something that activates ERK and AKT. But as you can see, there's a great deal of heterogeneity in the level of activation, not to mention a great deal of heterogeneity in what the cells were doing before they were stimulated. So this is the data that we're seeing, and this is a pretty common format to show the data in. So next, I want to talk just briefly about what different experimental situations we can kind of deploy this technique in. And there's kind of 4 that I'm going to talk about. You can be super creative with this and do a ton of different things, which is one of the things I love about the field. But the most basic one that I showed on the previous slide is just a signaling response. So you've got some cells, you look at them, and then you give them some drug or some growth factor, and then you look at what happens to them. Obviously, in each of these pictographs on the right, I'm just showing one cell, but we would have hundreds of thousands. So we can get a little bit more complicated than a single signaling response, and we can do multiple signaling responses. I alluded to this earlier. You could do 2, you could do 3, 4, whatever. However, there's also a lot to be learned by watching cells without stimulating them. It turns out that we've kind of started to learn that cells will, even when they're not perturbed, these signaling pathways can oscillate and pulse, and that those pulses and oscillations are actually related to things like proliferation. So you could see data like this even in the absence of stimulation. And then lastly, I just want to highlight that since we're doing video microscopy, we can see things other than the fluorescent signal. We can also see discrete events like cell divisions or cell death. We can track cells as they migrate, or we can maybe even see cells as they differentiate. So in that case, you could connect the signaling pathway activity that you see with then some later event like cell division. Okay, so with that, I want to just mention a couple different paradigms of cellular heterogeneity. I've got a very simple graph here showing 3 cells responding to some growth factor. They all started in the same place and they responded very differently. So there's at least 2 ways you could think about this, and there's a lot of gray area in between. But one paradigm kind of says that this heterogeneity is due to noise. And I'm going to leave noise a little bit undefined here, but what we do know is that all of these processes, the kinase activation inside the cell, it's mediated by chemical reactions, and chemical reactions are inherently stochastic, especially when there are low numbers of molecules involved in the reaction. And so if you think about a gene, there's only 2 copies of a gene in the cell, right? And so one school of thought sort of attributes the heterogeneity that you see in cells responding to noise. Basically, cells are doing a bad job of interpreting their environment, and so some of them are overreacting, some of them are underreacting. Now, another paradigm is heterogeneity due to pre-existing state. And so in this case, you imagine we've got these 3 cells, and we can only see one thing about them. We can only see their kinase activity. But it turns out that there's some other protein, there's actually hundreds of thousands of other proteins in the cells that we can't measure, but that is very different between each of these 3 cells. And so if you imagine this black protein is responsible for amplifying the kinase activity that we see here, if we could measure this, we would know in advance that the red cell is going to respond really strongly, the green cell is going to respond barely at all because of this pre-existing difference. And so this is not really noise or stochasticity. It is in fact very predictable. We're just missing the information. So on this next slide, I'm going to sort of summarize what I'm going to be talking about today. And so I'm calling this creative analysis of single cell signaling data because there's a bunch of different really cool stuff people are doing. And I'm going to try to, I'm going to talk in detail about stuff that our lab does, but I'm also going to introduce some other thoughts. So first off, we can think about mechanistic modeling of heterogeneity. And so this is a really good way to answer questions about where heterogeneity comes from, what molecular species might be responsible. And so these are just a couple stills from work from our lab from a previous PhD student named Phil Spinoza. And so I'll talk about that. Some other approaches include statistical modeling. And so here, instead of a mechanistic model, the authors, this was just a year ago in PNAS, they built a statistical model to try to understand where pulses, or try to understand the statistics of pulsing in the ERK pathway. And this is a little bit less mechanistic, but it's very easy to apply in different conditions, which is a huge benefit, as we'll see. Another approach is called information theory. This is a really classic mathematical theory or an electrical engineering theory, but you can also apply it to cells. This kind of considers that cells are trying to communicate, or the environment is sort of communicating with the cell, but that the ability of that communication is being corrupted by some noise. And finally, I'm going to talk about some dimensionality reduction, which kind of answers questions about how much heterogeneity actually exists. Probably a lot of people are familiar with techniques like PCA or other dimensionality reduction techniques. It turns out that's kind of difficult to use on temporal data. So we'll talk a little bit about strategies for temporal data. Okay, so first I'm going to talk about mechanistic models of heterogeneity. Again, this is work that was done in the Linderman and Luker labs at the University of Michigan. A lot of it was by a previous PhD student, Phil Spinoza. And so basically what we observed, I'm not going to get into these observations, but we had questions about heterogeneity in metastatic potential or in migration of cancer cells. And as we were trying to answer those questions, we kind of had other questions about where heterogeneity in signaling comes from. So this is sort of an overview of our experimental setup. We're measuring ERK and AKT dynamics using kinase translocation reporters. We're focused on ERK and AKT because those two kinases regulate migration and metastasis. And we're focused on activating those two kinases using the CXCL12, CXCR4 ligand receptor pair, or the EGF, EGFR ligand receptor pair. And so over here, I'm just showing a chymograph illustrating the data and the level of heterogeneity that we see when we stimulate cells with CXCL12. So you can see in these cells, AKT has a wide variety of starting activities and a wide variety of responsiveness. ERK is more on in these cells, but you also see it's got quite a bit of heterogeneity. So we saw all of this and we were kind of wondering where it comes from and why, whether there might be some reason why some cells respond more or less strongly than others. So Phil built this ordinary differential equation model that includes receptor ligand dynamics. Those are up here at the top. It's got interactions between a bunch of proteins. The black lines are going to be positive interactions. The red lines are going to be inhibitory interactions within this kinase signaling network. And it also includes ERK and the ERK KTR and AKT and the AKT KTR. Including the KTRs here is really nice because it allows us to actually predict what the observed dynamics would be in individual cells. So we calibrated this model and I'm showing just two different model curves here. So this is sort of one set of parameters. You can see the AKT KTR turns on strongly, whereas in this other set of parameters, the AKT KTR doesn't turn on at all and maybe even drops a little bit. So this was useful for understanding the sort of the dynamics that we saw, but what we wanted to do was fit single cells. And so we had this hypothesis that some model nodes or some proteins in this network could be affected by other cellular processes that weren't accounted for in the model. And so there are three of these that we took into account. I'm just going to use mTORC1 as an example. So we kind of thought, okay, mTORC1 is a broad regulator of metabolism. It's a regulator of the cell cycle, and it's affected by a bunch of other receptors, which aren't included in our model. And so in that case, if you had two different cells, they might be in different places in the cell cycle or have a different metabolic state, in which case you would expect that this mTORC1 node would sort of affect the cell differently. It would inhibit ERK and AKT activity to a greater or lesser extent. So we applied that same logic to RAS and PI3K as well. They're also pretty ubiquitously shared amongst a bunch of ligand receptor signaling pathways. And what we hypothesized is that heterogeneity in these three signaling nodes could account for heterogeneity in the ERK and AKT responses that we observed. So we created a library of model runs. And then what we did is for each experimental cell that we observed, we fit the best model run with it. And so I'm going to talk more about that on the next slide. So you can see here that we've got in a breast cancer cell line, we've got now 4 columns in our chemograph. So we've got the AKT, KTR in these left 2 columns, and there's the same experimental data you saw previously, now paired with a model curve immediately next to it. And similarly, we've got the same experimental data from ERK paired with model curve next to it. So you can see that the model that we built recapitulates heterogeneity in both AKT and ERK and can fit sort of the whole range of observations that we see. The other thing that I want to point out is that since each of these model curves is represented just by 3 parameters corresponding to noise or to external factors in RAS, PI3K, and mTORC1, we can put each of these experimental cells in a 3D space, right? So if we imagine we've got a 3D space kind of like this, each experimental cell will fit somewhere in this space. So this space actually also proved to be pretty useful for us. I'm just going to show 2 dimensions. I'm cutting out the 3rd dimension, the RAS parameter, because it's less interesting here. But if we think about the space, there are a couple things going on. First off, we can think about that there will be heterogeneous behaviors within this space. So if you're a cell in the bottom left, you're going to respond really strongly in AKT, whereas if you're a cell in the bottom right, you're going to respond really strongly in ERK. And these yellow and blue contours sort of show stronger, weaker AKT and ERK activity. Then what we can do is we can overlay experimental cells in this space. And so these are the contours that you can see. And what this shows is that some cells down here in this tip will respond very strongly in AKT, whereas other cells up here will sort of not respond in AKT or ERK. And based on our model is predicting that that's due to differences in how much mTORC1 and PI3K is sort of pre-existing in an active state in these cells. So I'm not going to dig too deep into this now, but feel free to ask questions. But what we found is that the cell state was repeatable, it was controllable. So if we perturbed the cells before we stimulated them, we could sort of we could move this contour around. And it also predicted some interesting ERK and AKT drug responses. So the other thing that we hypothesized is that the state variables that we're proposing explain the heterogeneity are actually not a function of the receptor. So I've got this simplified signaling diagram here. The thing to note is that RAS, mTORC1, and PI3K are inside the cell. They can regulate receptor expression, but we reasoned that different receptors that signal to these pathways should sort of similarly be affected by whether a cell has a lot of mTORC1 pre-existing or not very much mTORC1. And so to test this hypothesis, we took a different approach. Basically what we did is we stimulated cells with CXCL12 and we inferred their state variables. So that's identical to what we saw on the last slide. And you can see some more results from that down here. This is with a different cell line, but again, we've got experimental and model AKT and experimental and model ERK. And the fits are, again, pretty good. The data is a little bit noisier, so it's not perfect fit. But then what we were able to do is we were able to predict the distribution of EGF responses that we had. And EGF is going to have different ligand receptor kinetics, but again, we hypothesized that it would be affected in the same way by these extrinsic noise parameters. And so then when we do that and we test EGF responses, we do indeed see that the same set of the same distribution of sort of external model parameters was able to recapitulate the same dynamics that we see in when we stimulate with EGF instead of CXCL12. And so this sort of further gave us confidence in this idea that cell state was pre-existing in cells and could explain variability in multiple receptors. Okay, so this was one approach and it was useful, but I'm going to talk about a couple others. So I mentioned this previously, but some statistical modeling of signaling has also been really interesting. So there's been this observation for several years now that cells at rest will just pulse. They'll be doing all sorts of different things, but one of the things that they do is they pulse depending on sort of how likely they are to proliferate or to migrate. And so this is just showing some data from 2013. Cells cultured in 2 different concentrations of EGF, a low concentration and a high concentration, you'll see these sort of stochastic pulses. And so some work from last year tried to model these pulses as a self-exciting coupled point process. And so what that means is that if you think about the frequency of pulses in an individual cell, lambda, you're going to have some baseline pulse rate, and then there's going to be a function of the state of the cell. So if you just pulsed, you're more likely to pulse. And there's also going to be a function of your neighbors. So if your neighbor just pulsed, you're also more likely to pulse. And so I'm going to talk a little bit more about that on this slide. The simplest model you can imagine is one where you have a constant intensity or a constant probability of pulses. And so if you had observed dynamics that look kind of like this, your conditional probability of seeing a pulse would not change at all, right? It's just going to be a constant. A slightly more complex model that better fits the data that they observed is a self-exciting process, which basically just adds a couple more parameters. And it says that if you pulse, you're more likely to pulse again. There's some increased likelihood that's parameterized by some parameter A, and then there's some duration for which that effect lasts, which is going to be parameterized by time B. And so your conditional probability of pulsing is going to look more like this, where the history of the cell sort of affects its probability of pulsing again in the future. They added one more term to this, which is, as I said, it's not just self-excited, but it's also excited by neighbors. And so you can also have, this neighbor cell that's within some distance that's also affecting your cell. And so they kind of describe their parameters up here. So for some, if you have a higher spontaneous pulsing rate, you'll see more pulsation. If you have a higher self-excitation, you'll see more pulses back to back to back. And if you have high coupling, then for two neighbors, you'll see them be very synchronized. And so this is really cool because in the absence of any sort of mechanistic model, just given signaling pulse data for cells and their locations, you can infer these 5 parameters, your baseline rate of pulsing and your self and neighbor excitation parameters for any condition. And so this is really amenable to like a high throughput screening approach where you're doing a 96 volt plate or 384 volt plate, and you're collecting data in a bunch of different conditions, and you're able to sort of compare how those conditions affect some of these parameters. So I'm just going to show one result from this paper. This is showing the effects of perturbation using a drug called TAPI1. TAPI1 interferes with cell-cell communication. And so you would expect that it would decrease sort of neighborhood coupling. And so what they found is shown in these 2 graphs on the right. As you increase TAPI1 concentration, your baseline pulse rate goes down, but your neighborhood coupling also goes down. So this is, I thought, a pretty interesting approach. It doesn't require the mechanistic detail that some of our work required, where you're describing every reaction inside the cell or inside your model, which is pretty time consuming and pretty difficult. So this is another, I think, pretty interesting approach. So next, I'm going to talk about cell signaling as a communication problem. And so I'm just going to pull up that. Same schematic of ligand receptor communication from the past slide, and just kind of want to walk through some of these components in terms of like information theory and in terms of communication. So information theory is basically going to quantify how many different inputs we can observe or we can distinguish between in a population of cells. So if cells are acting as a switch, we're only ever going to be able to see 2 different inputs. They're either off or on. If they have a continuum of states, maybe we can see more of what the input is. And so we think of the information source as being the concentration of ligands in the environment, or it could be a ligand, it could be something else outside of the cell. We've got this communication channel that's formed by the chemical reactions inside the cell. There's some destination where this message is trying to go. That could be kinase activation, it could be DNA or gene regulation, it could be migration. In this case, we're going to consider just kinase activation. And finally, there's going to be some noise source, which in this case is going to be limitations in the ability of these signals to transmit a message. So again, due to that stochasticity we talked about, there can also be other chemical effects that could prevent a cell from sending sort of a perfect message. So I want to talk a little bit about quantifying cellular information transfer. The units we use are bits, and so that's going to be the log base 2 of the distinct outputs we can observe. So if you've got 2 bits of information, that means you can see 4 different outputs and so on. And so the way that these experiments are commonly set up is you can imagine you've got 4 different dishes of cells, and you're going to expose them to 4 different concentrations of some stimulus. In this case, it's EGF. And so if you have perfect communication, you might get an output something like this. So each of these graphs is showing ERK activity as a distribution. And as you move to the right, you're going to have higher ERK activity. In this case, I can be very confident. If I observe a certain ERK activity, I know what concentration it corresponds to, right? So if I see an ERK activity down around here on this pink distribution, I know that that came from 100 nanogram per mil stimulus. Now, this is not what we commonly see. Usually we're going to see something more like this, where these distributions overlap. And so you can kind of see that if you have larger variance or a smaller dynamic range and you start to see overlap, the ability to distinguish what input led to these outputs is degraded. And so in this case, maybe your mutual information is actually closer to 1 bit instead of 2. So people have deployed this strategy in a variety of ways. One really interesting approach, this is from a paper from 2014. They were interested in how cells, so there, I guess for some context, previous work had showed that cells were really bad at understanding their environment, and that the information processing ability of cells was like less than 1 bit, which is surprisingly low because you're a cell, you want to understand your environment so that you can make good decisions. And so they were trying to sort of improve upon that or understand where that comes from. So this is the data they measured at a bunch of different EGF concentrations. They got these distributions of ERK activity, and you can see these distributions are pretty heavily overlapping. So if they used any single time point measurement, yeah, cells have problems understanding being in a petri dish. That is true. But yeah, so if you have, if you look at any single time point, the mutual information of cells is very low. It's less than 1 bit. What they found here is that if you actually consider the whole, if you actually consider ERK dynamics over a whole time span, so a whole hour, you actually get much better mutual information. And so this graph is showing that as you use more and more time points to calculate mutual information, you can reach a much higher level of information transfer. And so this over here on the left corresponds to just using 1 time point, so any of these points on the graph. But over here at 15, that's using 15 different time points. You can actually start to distinguish 3 or almost 4 different inputs. So this was a pretty cool approach, but there's sort of an assumption here that all of the cells are the same. They're in the same state, and all of their differences are due to noise or randomness. That kind of it conflicts a little bit with some other assumptions about cells being in different states, right? And so another way that you can do that you can measure information transfer is to expose the same cells to different concentrations. And so on this slide, I've got I'm contrasting a different experimental approach that was used just a couple of years ago. And so in this, instead of 4 dishes exposed to 4 different concentrations, they had 1 dish of cells, and they exposed that dish to more than 4, but in this case, they exposed the same cells to 4 different increasing concentrations of a stimulus. The stimulus in this case is acetylcholine. They were studying calcium signaling. And what they found is that different cells actually have markedly different behaviors. And so what you see is that as you increase the dose for this black cell, it responds very quickly and goes sort of to the maximum of all the cells. Whereas this pink cell, it takes almost a 10-fold higher dose of acetylcholine to start to respond compared to the black cell, and it doesn't reach the same sort of maximum. And so what they were able to do is calculate mutual information between the dose or the response and the dose in individual cells. And they were able to extract a distribution of information capacity where some cells are way up here. If you have 3 bits of information, you can distinguish 8 different inputs, which is quite a bit higher, obviously, than what we saw previously around 1 bit. And cells actually have a wide range of sort of abilities to perceive their environment. So this again sort of gets back to that idea that cells maybe have a pre-existing state that can bias how good they are at understanding their environment. Okay, so on this slide, I wanted to got some animation issues, unfortunately, but on this slide, I wanted to just talk about 2 different dimensionality reduction techniques. So on the left, we're talking about functional principal component analysis, or FPCA. We're probably somewhat familiar with the idea of PCA. You've got a high dimensional set of data, and you want to figure out what the key sources of variance are in the data. So you use FPCA to identify the principal components that explain most of the variance. That doesn't really work for dynamic data because in dynamic data, you have to respect the fact that it's temporally ordered. And so this was from a group, Peter Sorger's lab at Harvard. They used functional principal component analysis to do dimensionality reduction on some signaling data that they acquired, where they exposed cells to different growth factors. And so you can see different example trajectories from 3 different growth factors here on the left. What they're able to do is perform functional principal component analysis. And what that does is it identifies that in their data, which includes hundreds of thousands of cells, these 3 harmonics can be combined in different proportions in order to explain most of the variance in the data. And so if you just if you stratify cells or if you quantify cells based on their first or their second and third functional principal components, they cluster into these nice groups based on their different dynamics, where cells that are up here in blue have much stronger transient dynamics. Cells that are red down here respond strongly and don't have that same transience. I'm also mentioning this just because I know I'm talking to a modeling group, and there's also some really cool connections between FPCA and sensitivity analysis. That's also mentioned in the bibliography link. So another approach to clustering or dimensionality reduction of high dimensional dynamic data involves using neural networks. And so in this case, this is from a group in Germany, the Olivier Pertz lab. They took cells and exposed them to 6 different growth factors. Conveniently, they're the exact same growth factors that were used in the paper on the left. They collected dynamics over quite a long time period, like 15 hours for both ERK and AKT. And then what they did was they asked a neural network to find features that could best classify cells into these different states, or could classify them based on what was perturbing them. And so they were basically they were taking the opinion of, we don't know what features matter, but a neural network might be able to find something interesting. So what you can see is that when you project those neural network learned features and do dimensionality reduction on them, you can get sort of different clusters of cells. And you can look at some of those clusters. So in this case, I'm just highlighting clusters 3 and 4, and you can see sort of typical motifs or typical signals that were in those clusters. And so this was also, I thought, a pretty interesting or pretty cool approach to classifying this dynamic data. All right, so I'm running a little bit long, sorry. So I want to conclude by talking about a couple different things. We kind of had this spectrum of how you explain heterogeneity. At one end, you could say it's all due to noise or randomness. At the other end, you could say that cells are in a pre-existing state and then they respond perfectly based on that state. And so what we kind of see is that information theory attributes a lot of heterogeneity to noise. The statistical modeling approach we talked about is kind of in between, and then we've got this mechanistic modeling approach where we don't invoke noise at all, and we just use pre-existing cell state. So kind of depending on where you think your heterogeneity is coming from, you might want to pick one or the other. I want to just talk briefly about what we kind of see as the clinical utility of cell state. I had this, I showed this figure earlier, and then I've drawn a much cruder representation of it using PowerPoint here. But if we imagine we've got a pre-existing cell state that we can learn using modeling, we might be able to target different subpopulations of this cell state with different drugs, if we know what's different about cells that are up here and down here. Another approach that we might use is maybe we can manipulate the cell state and give some drug that pushes all of the cells out of this bottom left corner and into this upper right corner, and then they can all be targeted by the same drug. Yeah, so we're in the Linderman and Luker lab. We're trying to utilize this in a bunch of different ways. We're looking at different inhibitors, the effects of different tumor microenvironment variables, and we're also sort of starting to study proliferation, migration, drug resistance. And so we kind of think that by understanding sources of heterogeneity, we'll be able to better treat cancer. I just want to acknowledge former PhD student Phil Spinoza and Ray Asari. I used some content that we had made together for this talk. Gary and Kathy Luker Lab and Kenneth Ho, and then we're funded by NIH, some internal funding at U of M and the Keck Foundation. And I am very long, I'm so sorry. But yeah, so I'll just leave this slide up and I'll take some questions. And yeah, thank you very much for the opportunity. Also really appreciate Joel for reaching out.
Thank you for a great presentation, Patrick. Questions from the audience?
Yeah, I have a question. Hey, Patrick, thank you very much. You did an excellent job providing the bibliography if we want to go more in depth with it. And but one of the questions, my background is not biology. Do you
Okay.
notice if the heterogeneity is capped by the, the parent cell to the child cell whenever there is a cell division, or do you notice that they're way more open to mutations?
That is a very, very good question. What we find is that, I guess, the maybe unsatisfying answer is it depends. So what we know is that cells can divide in different ways. So you can see cells asymmetrically divide or reproduce, or symmetrically divide. And when they reproduce, some elements of cell state can be transferred to daughter cells. Some of them can be sort of reset. And so to give one example that we sort of studied, or the Luker lab has studied, mitochondria are these really cool dynamic organelles that are responsible for generating energy. So sometimes when cells divide, all of the new mitochondria in the cell will get sent to one daughter, and all of the old mitochondria, which are less effective, will get sent to they'll get sent to a different daughter cell. And so in that case, those two cells are inheriting a very different state. In other cases, though, we can see, oh man, okay, so there's another paper that I didn't mention here and didn't make it into the bibliography, but it should have, where they were studying AKT activity or something immediately downstream of AKT as it, they were doing sort of the same approach where they gave an incrementally increasing dose of insulin. And what they found is that daughter cells had highly correlated abilities to, so they had, I'll just go back to it. They had graphs like this one in the bottom right, where they were studying mutual information, and they found that daughter cells, if you, like you and your twin or like you and your sister cell were much more likely to both have similar levels of mutual information. And so that says that state is inheritable. That was a lot of words, I'm sorry, but it's a really good question. It's just also a really complicated question.
It is great. It is great. Thank you.
Patrick, that was a great talk. Could you skip back to about slide 12 or so, somewhere back there?
Yeah, totally.
So you're invoking, to a certain extent, other signals impinging on the cell. So your mTORC1 signal is you modify it, saying that there's some other signal contributing to it. So we're gonna pick some random distribution and tweak mTORC1 up and down a bit. It could also be just differences in concentration of mTORC1.
Yeah.
Or indeed difference in concentration of everything in the system. But I mean, it gives you the same functional result. You're basically just making the cells not be identical to each other. But in any case, my question is, how much do you tweak mTORC1? What is the distribution of values that it can have based on this external variation?
So the variation in concentration is about, it's on the order of like 1 log of difference in concentration. And yeah, you're absolutely right. It could be cell, sort of the concentration of the complex could also be different.
Okay, but the big thing is, so you're doing a log, you're doing a pretty
Yeah.
big change. Okay.
Yeah. And it's different for each of these. It can be different for these. It can be different for each of these species, based on the rate constants around them in this model of ODEs. They'll be sort of, a small change can be drastically amplified by the system, or a large change could be attenuated by the
Yeah, so the mTORC1 has its own intrinsic sensitivity.
Yeah.
And, depending on what you pick in the model, you may get things that are very sensitive and things that are they're much less sensitive.
Right.
Oh, okay. All right, thank you.
Yep.
I have a question. Oh, Sorry.
Yeah,
Okay.
Amanda, go ahead.
And this slide is fine because it's about the cell cycle. Is there any way to kind of sync up all of the cells so they are synchronized at kind of the same point in the cell cycle, or is that already kind of being done?
So this slide is, okay, I guess I'll start by saying there are experimental ways to sync up the cell cycle where you give a drug that prevents the cells from cycling and then you take it away, and then every cell sort of starts the cell cycle at the same time. That's been done for a while. It's a blunt instrument, I would say. It can affect a bunch of things about this. Like, it's a poison, basically, so the cells are not really happy about it. Some other work, I'm thinking specifically of like Sabrina Spencer's lab at UC Boulder, has done that computationally, where they're doing video microscopy of cells as they cycle, and then they're sort of computationally syncing them up. So they're saying like they're sort of referencing every trajectory that they get to the start of the cell cycle, and that's led them to some pretty cool conclusions. In this particular work, I'm invoking the cell cycle here as a one of the many things that could cause variation in mFORC1. But because of how we do the image processing, we actually we threw out cells that were actively cycling. Like, we didn't have cells that were dividing during the experiment. So it's not, I don't know. We don't know for a fact, like, well, I guess what I'm saying is, like, we didn't test whether synchronizing the cell cycle mattered, but we also like had some reasons to think that we're sort of artificially reducing the heterogeneity that you see in the cell cycle, if that makes sense.
So this is John. I'm not a biologist. I don't think, there's lots of this stuff that I don't have any idea what you're really able to see. But I'm making an assumption. I have a community of cells. Some are the same family, others are different families of cells, and they're all mixed up somehow. And for life to go on, there must be some reason to have to communicate some things.
Mm-hmm.
Do we know any messages? Are there like use case, and it's, excuse my awkward kind of questioning, but that the cell says something just happened to me, and I have to tell the other cells that happened. And there's
okay.
a reason that they have to know. And therefore, when that happens, it causes me to create and put out some molecule that is received by the other cells. Why?
Yeah, so I'm really, okay, so I knew that this was a virology talk, so I found a couple of papers about that, and I mentioned one in the bibliography that I sent out. So there's this really, really cool study that I think is related, and I'll mention it, and then feel free to ask other questions. So basically, what happens when cells are infected with HSV1, so a specific virus, is that macrophages will secrete, macrophages being immune cells, they'll secrete a molecule called TNF, or tumor necrosis factor. And what they found, and so that's sort of a known thing. TNF is associated with a type of cell decision-making called apoptosis. Apoptosis is controlled cell death. Sometimes cells will get too old, or they'll be sort of injured somehow, and it's better for the organism for those cells to essentially kill themselves or go through controlled cell death. So what they found in this study is that when there's no immune challenge, when the immune system is not active and there's no TNF in the system, cells do a very, very good job of making apoptosis decisions. So, but they also are slow to make them. What they found is that when you infect cells with HSV1, macrophages then secrete TNF, and that TNF causes cells around the macrophages to, instead of make slow but accurate death decisions, they make very fast but much less accurate death decisions. And so what I'm trying to get at here is that that's one example where one cell type is getting a stimulus, and that stimulus is the presence of this virus, HSV1, and it is then communicating a sort of a set of instructions to another cell type, and that other cell type is just sort of the epithelial cells that are around, in order to control how those epithelial cells make the sort of the programmed cell death decision. So that's just one example, but does that kind of get at what you were asking, or am I maybe just blabbering about stuff that isn't helpful?
Why would they speed up?
Um,
What would be good about getting the message to the other cells that you need to speed up some process or something?
Because if the cells, that's a great question. If the cells are infected, if they kill themselves quickly, then the infection, the virus can't grow inside of the cell. And so if you make that decision very quickly, then you're going to limit the spread of infection. But you can imagine if you're healthy, then you don't want cells to just accidentally be killing themselves all the time, because then the organism has to replenish the cells. Does that answer your question?
You know, it may be a little silly, but it almost sounds like it would be fun to get a big room full of tables with a few people of each table, and each table is a cell. And like, here comes the message. What's the message mean? What are we going to do if we get that message, and why? And to, you know, I don't think anything goes on in nature that doesn't have a purpose.
Mm-hmm.
There's no waste out there, and to understand and you know, to think of a cell as an intelligent thing that thinks about the messages it gets, and then something happens, and that combined with the physics level of the closer you get to seeing things that are consistent, you know, the idea that the reason that molecule is released is because there could be no other thing happen. I mean, that molecule gets released under those conditions because under the laws of physics, that's what has to happen. And then that molecule causes something. And if you think about the cell community of speaking different foreign languages, it's like, okay, you know, if you hear the message and you talk that language, you think about it. If you hear the message and you don't talk that language, something else happens. I don't know. It just would sound like a kind of fun thing to do.
No, I think that's, yeah, trying to get human beings to come up with sort of like, what's the most effective way you can communicate a message given these rules? And the rules are pretty unforgiving laws of physics that have to do with the intrinsic stochasticity of these reactions. Like that, yeah, that could be pretty cool.
Yes, I'm not thinking random humans at the table. I'm thinking of people who understand some particular cell really, really well. And okay, anyway, thank you very much.
Yeah, of course. Good question.
So I've got a question for you too, Patrick. So with this cell heterogeneity, and when we think about mechanistic models, when we build our mechanistic models, we use, you know, published, you know, in vitro experiments as a way to validate our mechanistic models, right? And these validations are not always great because many times of this heterogeneity of the in vitro experiments, right? There's do you know, then you have a challenge of, do you average out the in vitro cell response and average out your simulations of the model and compare those, or, you know, so this creates a challenge in terms of validating your models, not just understanding the heterogeneity of the cell response.
Yeah, I don't. You're absolutely right that that's a huge challenge. I don't have, I think, a great answer of like, given this heterogeneity, this is what we need to do. I will, I guess, some things that this heterogeneity can allow you to do is it can allow you to see, for instance, your dynamics could be totally great because all of the cells sort of follow the same dynamics. If you're calibrating a dynamic model to a collection of cells, in a lot of cases, like sometimes you'll see that the cells, a lot of them do look like the average cell. And so studies like this can allow you to say at least like, is it plausible to calibrate to averages or to calibrate to sort of an average cell? In other cases, you might see that in fact, like the process is bimodal or there's multiple populations. And in that case, you would need to sort of calibrate to different subpopulations. But I mean, I guess so the philosophy in the Linderman lab has always been like, what is the simplest model that can provide us with like useful insights? And so I guess the other answer might just be that the closer modelers are sort of integrated with experimentalists, where you can figure out what those useful experimentally actionable insights are and then try to acquire them. Like I don't think that this is a unique, it's not like this is the first problem with ODE models of cell signaling, right? There are other problems too. And so I guess maybe that's another answer is we already know that ODE models are not perfect. This is just one other layer of imperfection. And so just getting to those experimentally actionable insights is still sort of the key. I don't know. That could be a pretty bad answer though. It's a really big challenge.
I have one question. Have you tried or have you read about someone that tried to combine models as you were showing here, the single cell fitting, considering the way you did, and probably like the exterior part of the cell being more of like a probabilistic or a information theory based approach? Do you think that is plausible? Is it possible? Do you see any change?
Okay, so people in this group are probably somewhat familiar with like the Gillespie algorithm, where you have a set of reactions. It's very similar to an ODE model, but you can explicitly simulate the distribution of the stochastic distribution you're going to see. There has been work where people have created signaling models, and then they have compared whether they've run those models using the Gillespie algorithm or using just ODEs, using sort of a smoothing. And what you tend to find, I'm thinking of two papers in particular, what you tend to find is that when you get to the number of molecules involved in mammalian cells, the influence of true stochasticity goes down pretty dramatically. And so that's one answer. I have not, to my knowledge, I don't know of any models where like there's a sort of a stochastic element outside of the cell and then sort of an ODE on the inside of the cell. I mean, I guess one example would be the agent-based models we build, where we've got agents that are immune cells and they're moving around stochastically, which is changing the environment. But then there's some reactions that occur deterministically within the cell. And another, this is going to be a little bit speculative because it's not my area of expertise, but I know that people do talk a lot more about stochasticity in gene expression. That's not something I'm really getting into here, but that's maybe another place where you could see models where there's a sort of a differential equation section that's then sort of merged with like a stochastic section. But again, I don't want to speculate too wildly.
Thank you.
Yeah.
I had another question that's similar to kind of what John was asking before.
Mm-hmm.
So it seems a lot of the signaling experiments are done where the ligand is kind of uniformly distributed across the cells. And I know you showed some experiments where it's just like different concentrations and they have delayed or different responses. So there have been experiments where it's more of of one location where the ligand is high concentration, and then there's a gradient kind of going off of that?
Yeah, I'm actually, okay, sorry, you're gonna see all of the slides that I put together while I was making this. But I actually have a really cool example of that. It is,
And
okay,
then these are all the same cell type,
yes,
although
yep,
heterogeneous, but still the same cell type kind of there, right?
yeah,
Okay.
yeah. So you'll start to see people do what are called like co-culture. I guess just to address that last question, sometimes you'll see people do like co-culture experiments where you have two different cell types, and that also gets back to the question that John was asking, where it's like the cells are communicating. But okay, so you were originally asking about sort of like a spatial component of
Yeah.
the stimulus. Yes. Okay, so in my initial version of the slides, I was going to talk a lot more about what inspired us to look at heterogeneity in ERK and AKT, and now I guess I'll just do that. So this is an older paper. It's from 2014 in the Luker lab, and it's a little bit hard to see, but this is a microfluidic device that's got three components to it. So on the left side, you've got cells highlighted in blue. Those are acting as a sink for a gradient. In the middle, you've got cells highlighted in green expressing CXCR4, and that's a receptor that I talked about. And then on the right, you've got red cells that are expressing CXCL12. And so that is a chemotactic ligand. It's really important in like organizing the immune system. It's also involved in like cancer metastasis. So the way that this microfluidic system works is that CXCL12 cells secrete this ligand CXCL12, and then CXCR7 cells absorb it. And so you get this gradient that forms that's represented by this red triangle at the bottom. And so in this case, we can actually see that cells, they're exposed to a continuous stimulus, but it's got a spatial gradient to it. And so what we saw is that cells migrate towards the red cells. They migrate up the gradient, which is kind of what we expected to see. What's really interesting is that a bunch of cells exposed to the same gradients that are isogenic, they're the exact same genetically. A bunch of cells don't migrate at all. And so that was kind of what inspired this whole mission. So this is one example. This is relatively old. We've come a long way since then, and it turns out that you can actually buy microfluidic devices from a company called Ibidi that will impose a gradient on cells. And so you can actually create this gradient instead of having a custom microfluidic device, you just buy something. And so that's something that we're starting to do experiments with. Kenneth, the postdoc that I mentioned in the acknowledgement slide, he's been doing a bunch of experiments where he looks at cells migrating under a gradient. So does that kind of start? It's still like kind of the simplest possible situation where it's like just a one-dimensional gradient, but it is like some spatial organization, if that makes sense.
Yeah.
I get this picture of a fire in a theater where one of the cells yells fire, and then everybody goes crazy. Everybody starts yelling. The messages are all over the place, but it's chaos.
Yeah.
How does the cell community manage that?
That's an interesting analogy. That kind of does remind me of the like immune example. And there are cases where if the poison or if the problem that the cell is trying to address is serious enough, you would want one cell to be able to yell fire, and then all of the cells sort of start to die or start to apoptose. Obviously, we don't want that for humans. I will also say one of our sort of theories is that the distribution of cells, the heterogeneity that we see in the cells, it has a survival benefit. And so I guess I don't have a great answer to the question of like how cells manage the chaos. I guess I would just say that's something I'm really excited to be researching. Yeah, I don't think we have a good answer yet for how the cells manage the chaos. And I think a big part of that is that we don't have great ways of really like simulating experimentally all of the different inputs that are going into a cell. And I guess that sort of opens up new experimental approaches where you're trying to measure heterogeneity in different ways or control cell environments in different ways. But it also opens up new modeling approaches where you're trying to see, like, is how can a cell make sense of all of the different inputs it's getting, and modeling different ways that it might be doing that. So again, it's a good question, but I don't think anybody knows right now.
I had one last question. It's not related
Totally.
to viruses, but at the end you mentioned that you guys were interested in it for cancer. Is it more for cancer treatment, kind of after the cancer has initialized and began, and it's more of a drug component, or is it a little bit more? Are you guys looking at it also for the cancer prevention, a little bit of like seeing this as the potential of we can maybe use this to see if cancer will or will not come from these cells? Is it more of like a treatment, or is it more preventive?
More of a treatment, I would say. And so the specific specialty of the Luker lab is studying metastasis that's mediated by CXCR4 and CXCL12. And so the real idea is that when metastasis happens, obviously that's localized cancer is fairly manageable. It's not good, obviously, but it's fairly manageable. And so when cancer starts to metastasize, that's when it becomes a problem. And so the sort of the real question that the Lukers are interested in is what makes a couple cells metastasize while the rest of the tumors stays behind. And so the hope of, well, some of the hope of my work is more related to just straight up treatment and like drug resistance. But I would say kind of the goal is to identify what specific conditions will lead to metastasis and how can we like prevent those conditions from forming. So it could be an environmental factor in that gradients form in a certain way within the tumor, or it could be a cell intrinsic factor where cells in a certain niche surrounded by some stromal cells and some immune cells are primed to migrate, something like that. And the goal is to find specifically why those niches form and then stop them from forming so that metastasis doesn't happen. Does that kind of answer your question?
Yeah. Thank you.
Yeah. No, thank you. All of these are really, really great questions. Oh, cool. Sorry, Tom, I'm just clicking on your paper. Oh, this sounds very cool.
This was that work was purely through modeling. It was a large scale mechanistic model of signal transduction. Actually, I still remember trying to reproduce the phenomenon of sustained ERK response versus just a transitory response as a phenomenon to validate the correctness or accuracy of the model, and it was very difficult. So,
gotcha.
but
yeah,
this was purely computational. We didn't collect
Okay.
any data to like you're doing. Yeah.
No, this is very, very cool. Thank you so much for sharing. Yeah, this is.
You mentioned in the end, if I just can pick one last one from you, you mentioned at the end using drugs to force a certain type of cell as a treatment and reducing heterogeneity. Do you know a specific, or do you know a specific drug that does that, or is that just speculative for now and is like a future?
It is currently speculative. It was a good way to end the review, so that's what I went with.
No, it's great.
So I guess I will say it's not like we have not translated our understanding of cell state into a rationally designed like chemotherapeutic regimen by any means. This is my bad. I should have looked over this, but there are examples of rationally designed chemotherapeutic regimens where you give one drug and then it sensitizes tumors to something else. So it's not fully speculative to say that such a thing could be done. It's just not, we haven't gotten there by any means. So yeah, that's totally a good question though. Oh, I don't think I put my contact information anywhere either. If people want to email me, I'm putting my email in the chat, and I'll also put it in that bibliography document. But yeah,
Thank you, Patrick.
yeah, of course. Too, and yeah, do people have other questions or ideas or suggestions? Be too happy to chat.